Welcome to our very last section in Unit 6. We're going to talk today about nucleic acids. So if you'll recall, we have been looking at different organic compounds, 4 of them exactly. We've talked about carbohydrates, lipids, proteins, and now the very last one, nucleic acids. So what is the function of nucleic acids? You've probably heard some of these words before, DNA and RNA. DNA in particular, probably you associate with genetic information. DNA and RNA are both nucleic acids. So given that information, DNA and RNA are responsible for dictating amino acid sequences of proteins. They determine that. That in turn controls basic life processes, and that's part of the source of the genetic information in chromosomes. That's where we get that. So we're going to learn more about those processes in a later unit, but for now, you just need to know that DNA and RNA are associated with genetic information, transferring that genetic information, and sequencing proteins. So the nucleic acid structure obviously has not always been known. As a matter of fact, it was in 1953 that 2 men by the name of Watson and Crick published an article that determined the shape and structure of DNA. Prior to that, no one really knew what the structure was, and schools actually taught that the structure was quite different than what we see here and what we know today. So Watson and Crick are responsible for discovering this structure and revealing that to the world. So what is the structure of DNA? Well, let's start with the name DNA. DNA stands for this molecule, deoxyribonucleic acid. So the D is right here. Underline that, the D, the N, and the A. DNA. What does that all mean? Wow, what a long word. Let's break it down, and we're going to start with the simplest part of the DNA. We're going to look at the DNA nucleotide, okay? The nucleotide is this portion here, and it has 3 parts to it. It has a phosphate group right here. These are the atoms involved in this phosphate group, phosphorus and oxygen. It has a sugar. Remember talking about sugars. The sugar right here is a specific sugar. It's a deoxyribose. That's the sugar. And it's called deoxy because it has 1 less oxygen in it than simply ribose. It's 1 fewer. So it's called a deoxyribose. And the third component of this nucleotide is a nitrogenous base. It's a great big word. It means it's a base that has nitrogen in it somewhere. It can vary. There are 4 different nitrogenous bases, and this can be any one of those. So let's take a look at what our choices are for a nitrogenous base. These nitrogen-containing bases, there are actually, there are 5 total. We're going to talk about, we're not going to talk about this one for right now, uracil, but let's talk about the other 4, because the other 4 are present in DNA. Cytosine, we represent that with a capital C. Thymine, we represent with a capital T. Guanine, we represent with a G, and adenine, we represent with an A. You can see these are categorized differently. These molecules are larger. They have 2 rings. You see that? These molecules are only 1-ringed molecules. So when we look at our DNA molecules, remember, our nucleotide sequence has a phosphate, a sugar, and then a nitrogenous base. And that nitrogenous base can be either cytosine, thymine, guanine, or adenine. And we simply shorten them by calling them C, T, G, and A. So we're going to put these together a little bit. Our structure is going to get a little bit more complex as we go along. So here, let me just point out what we have. We have our phosphate and our sugar right here. And then we have a nitrogenous base. Right here is thymine. So this is putting our molecule together in a more complete way. The phosphate sugar repeat. We have phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar. That repeats over and over. This is called a backbone. This makes up a backbone. Here is our nitrogenous base. Nitrogenous bases pair, and they always pair with the exact same partner. T always pairs with A. G always pairs with C. Right here, the G and the C will go together, the A and the T will go together regardless of what side they are on. And on this side, the sugar phosphate backbone is repeated going down the other direction. That's what we call anti-parallel. So while these sugar phosphates, the backbone over here is going up, these sugar phosphates are going down. They are also connected to a nitrogenous base, and these bases are loosely bonded with a weak bond called a hydrogen bond. So in between the bases, there are hydrogen bonds that hold them together. Now, let's step back a little more. Here we have our DNA molecule coming together. When we step back further and look at this whole molecule, it looks like this. An alpha double helix is what the structure is called. In between, here we have, right along here you can see, I'm tracing the backbone. That backbone, remember, is the phosphate sugar backbone, okay? And then this direction, our phosphate sugar backbone also, anti-parallel. And they are twisted around in what we call a double helix. In between those two backbones, we have our pairs. We have our, I'm not sure what the color coding on here, but they have the C and the G and the A and the T, and they are all color-coded. Remember, the T and the A always go together. So in this diagram, we should see orange and pink pairing up, which they do right here. Then we should see the G, the C and the G pairing up. Those are yellow and green, and those are pairing up right there. And they always pair in that same fashion. And in between, they are being held in place by hydrogen bonds. And this is our structure of DNA. So now, remember I mentioned another molecule, RNA. RNA is also a nucleic acid. And what does it look like? Well, remember DNA was deoxyribonucleic acid. RNA stands for something too. Stands for ribonucleic acid, and the letters are there, R, N, A, ribonucleic acid right there. So here is just our DNA molecule again. RNA is a simpler version of that. It's only a single strand. That is the main difference, but there is one other small difference, and that is that the nitrogenous base, it does not use thymine. It uses uracil instead. So in our DNA, when we had thymine pairing with adenine, in our RNA molecule, we will have uracil pairing, or uracil and adenine. They won't pair though, because it's only a single strand. We don't have this other strand, okay? So uracil is replacing the thymine. So let's compare and contrast DNA and RNA structure. Here's just a different representation of what we've been talking about, the double-stranded DNA and the single-stranded RNA. And that's our first main difference that is very obvious. In DNA, we have a double-stranded helix. In RNA, it's only single-stranded. Also, thymine is present in the DNA nucleotides versus uracil in the RNA nucleotides. In DNA, a deoxyribose sugar is present, is the sugar that's used, and that has one less oxygen than the ribose. In RNA, the sugar in the backbone is a ribose sugar, has one more oxygen than the deoxyribose. DNA and RNA both have the same sugar phosphate pattern in their backbone. Obviously, the backbone itself is a little bit different because the sugars are different, but they both have the same pattern in their backbone. Sugar, phosphate, sugar, phosphate. So those are the main differences between these two molecules. Advanced ideas. Here are some great ideas for you. You can read the article that was first published by Watson and Crick. If you cannot find it, ask me, and I have copies of those. What were the main structural components that they proposed for DNA? Or prior to their proposal, what was previously thought about the structure of DNA? What were teachers teaching students that was the structure of DNA? And what are the functions of DNA and RNA in the cell? You can go ahead and look that up. We're going to study that later on, but those would be ideas for advanced proficiency, and I look forward to seeing what you come up with.